Chromatography is an analytical technique used to separate, identify, and analyze the components of a mixture. It works by separating substances based on how they interact with two different phases. Because different substances travel at different speeds, they can be separated and identified individually. The principle of chromatography is based on the different attraction of substances to two phases, the stationary phase and the mobile phase. Components that are more strongly attached to the stationary phase move more slowly, while components that are more attracted to the mobile phase move faster. This difference in movement allows the mixture to separate into its individual components. The stationary phase is the part that remains fixed in place, such as silica gel or paper. It interacts with the sample components. The mobile phase is a liquid or gas that moves through the stationary phase, carrying the sample along. Together, these two phases make the separation process possible. Chromatography is important because it provides accurate and reliable identification of substances in complex mixtures. It is widely used in many fields, including pharmaceuticals, food safety, environmental testing, and forensic science. In our presentation, we will focus on its application in forensic science. Chromatography is used to analyze blood and urine samples to identify drugs, poisons, and alcohol. This information helps forensic science investigate cases involving drug abuse, poisoning, impaired driving, and unexplained deaths. With that introduction, I will now hand over to the next presenter, who will explain how chromatography is used in forensic science.
Today, I will present the application of chromatography in forensic science, especially its role in identifying drugs, poisons, and alcohol in blood and urine samples. For the background of the application, chromatography is an example technique used in forensic toxicology. It helps investigators to detect and analyze substances such as drugs, poisons, and alcohol in biological samples. This technique is useful in cases involving drug abuse, poisoning, overdose, and drunk driving accidents. It works by separating complex mixture into individual compounds, making identification easier and more accurate. Why chromatography is used? Chromatography is widely used because it has several advantages. First, it is highly sensitive, meaning it can detect even very small amounts of substance. Second, it is highly selective and can separate compounds with similar structures. Third, it provides accurate identification through retention times and other analytical data. Lastly, it can measure the concentration of substance in complex samples like blood and urine. Because of these advantages, chromatography is one of the most reliable methods in forensics investigations. These are the types of chromatography that have been used in forensic toxicology. The first type is gas chromatography or GC. It is mainly used for volatile compounds, which are substances that easily evaporate, such as alcohol and certain solvents. The second type is high-performance liquid chromatography, or we call it as HPLC. It is used for non-volatile and heat-sensitive compounds. This method is commonly applied in detecting prescription drugs, illegal drugs, poisons, and drug metabolites. And the third one, we have thin layer chromatography, or TLC. It is a simple and ineffective screening technique. TLC is mainly used for preliminary drug testing and poison screening before more advanced analyses are performed. You must be wondering, the theory of how separation occurs. The principles of chromatography is based on the interaction between two phases, the stationary phase and the mobile phase. Compounds that are more strongly attracted to the stationary phase move more slowly, while compounds that are more attracted to mobile phase move faster. Because different substances move at different speeds, they become separated and can be identified individually. For examples, in GC, separation depends mainly on volatility. In HPLC and TLC, separation is large based on polarity.
Alright, let's talk about methodology. When a forensic scientist gets a raw blood or urine sample, we can't just inject it straight into our instruments. The proteins and fats will instantly ruin the system. So step 1 is the chemical cleanup called solid phase extraction (SPE), which you can see on the slide. We pour the messy sample into a specialized cartridge. This cartridge acts like a chemical magnet, trapping our hidden suspects, the drugs or poisons, while letting all the biological waste wash right out. A final rinse releases our now crystal clear, concentrated sample. Now that it's clean, we have to separate the chemical crowd. Depending on the culprit, we take one of two analytical highways. For volatile targets like alcohol, we use gas chromatography. For heavy heat-sensitive toxins or illicit drugs, we use high-performance liquid chromatography, which is HPLC. As the diagram shows, a mobile phase forces our sample through a tightly packed column. Because different chemicals travels at different speeds based on their structure, they spread out. By the end of the line, a chaotic mixture is perfectly separated into pure individual compound. So what does the final data look like? When those separated chemicals cross the finish line, they hit a detector, creating a graph called chromatogram. We read this using two main clues. First, retention time. How long the chemical took to pass through the column. Since every drug has a signature speed, this identifies what it is. Second, peak area. The bigger the spike on the graph, the higher the concentration of the poison in the system. Pairing this with a mass spectrometer gives us an undeniable chemical fingerprint. The ultimate gotcha moment in a forensic lab.
So now we move on to the advantages of chromatography in forensic sciences. First of all, chromatography has high sensitivity, in which chromatography can detect very small amounts of drugs, poisons, or alcohol in blood and urine. And then, it has high accuracy and reliability, in which it produces reliable results that can be used as scientific evidence in court. And it also separates complex biological samples, because blood and urine contains many compounds and chromatography separates the target substances from other components. Chromatography can also identify multiple substances simultaneously, in which a single analysis can detect different drugs, poisons, and alcohol metabolites. Last but not least, chromatography is a widely accepted method in forensic laboratories, in which techniques such as GCMS, gas chromatography, mass spectrometry, and LCMS, liquid chromatography, tandem mass spectrometry, are considered the gold standard in forensic toxicology. Now, we move on to the limitations of chromatography in forensic science. First of all, chromatography requires expensive equipment, in which advanced instruments like GCMS and LCMS are costly. It also requires trained personnel, in which skilled analysts are needed to operate the instruments and interpret the results. Chromatography in forensic science also requires time-consuming sample preparation, in which blood and urine often require extraction and purification before analysis. Also, not every substance can be analyzed by one method. Some compounds require gas chromatography, while others require liquid chromatography. Last but not least, the results depend on proper sample collection and storage, in which poor handling may affect the accuracy of the analysis. In conclusion, chromatography is a powerful analytical technique used to separate, identify, and analyze the components of complex mixtures. In forensic science, it plays a vital role in detecting drugs, poisons, and alcohol in biological samples such as blood and urine. Its high accuracy, sensitivity, and reliability makes it an essential tool for forensic investigations, toxicology, and legal proceedings. Although advanced chromatographic techniques require specialized equipment and trained personnel, they remain one of the most trusted methods for providing scientific evidence and ensuring accurate forensic analysis.